So it appears that we are officially online. Hello, everybody, and welcome to another session of our Sussex Vision seminar series, as always within the Worldwide Neuro Initiative. I'm George Kafedis, a master's graduate from Thomas Euler's group, and currently a PhD student with Tom Baden. And as your host for today, I would like to once again begin by thanking Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest from Oregon Health and Science University and KCI Institute, Professor Benjamin Sivyer. Ben studied biomedical neuroscience at the University of Queensland. During his PhD in the lab of David Vaney, he focused on the ganglion cells of the rabbit retina, characterizing novel types and adding to the understanding of the function of local edge detectors, uniformity detectors, and direction selective ganglion cells. In 2011, he joined as a postdoctoral researcher at the lab of Stephen Williams before moving to Janilia and the lab of Gabe Murphy. He first joined Oregon Health and Science University as a postdoctoral fellow with Roland Taylor before starting his own lab there in 2017, if I'm not mistaken, and becoming assistant professor of ophthalmology. Throughout his academic years and his lab nowadays, they study the function of visual circuits and how that is disturbed and perturbed in disease. They employ a wide array of techniques from single cell and multi-electrode IFIs to transcriptomics and to behavior. And today we have the pleasure of hearing about their latest and I'm sure exciting findings in his talk entitled The Melanopsin Mosaic, Exploring the Diversity of Non-Image Forming Retinal Ganglion Cells. So without any further ado from my side, please all welcome Professor Sivyer. Ben, the stage is officially all yours.
Thank you so much, George. Let me just share my screen here. How's it looking now?
I cannot see it yet. Maybe you haven't screen shared. Would that be the case?
There we go. How about now?
Lovely. Yeah.
And you're still seeing it full screen?
Definitely.
Awesome. Thank you so much. So nice to meet you, George. And it's such an honor to be invited to give a talk in this seminar series. It was really cool to receive that invitation, and so thanks to George and thanks to Tom for the invitation, and thanks for coordinating this, George. I know it's a lot of work, and it's awesome to meet you. So today I'm going to give a talk about melanopsin ganglion cells and how they form a mosaic, and a specific subtype of these cells forming a mosaic in the dorsal retina, encoding light reflected in the lower visual field. I hope that my introduction will convince you that regional specialization of neurons is sort of conserved across many species, and it's not just a sort of niche in mouse retina. And this journey started with a chance discovery in a transgenic mouse line that has really opened a wonderful area of investigation in the lab, and formed really exciting new collaborations. And so I'm going to show you sort of a snippet of what we've been doing recently in the lab, some work that's published, some work that's unpublished, and then at the end I'll talk a little about some future directions, which I hope are exciting. But before I do that, I just want to acknowledge the wonderful people in my lab and their work on this project. Most of the work I'm going to tell you about today is performed by Mike Berry, who was an MD PhD student in the lab and is currently in applications for residency and doing interviews, and Tavita Garrett, who has helped out with this project. Many other people helped. Mark Meadows in the von Gersdorf lab and Misha Moldovan in Chuck Allen's lab helped sort of spearhead some of these synaptic recordings that we did. And then others like Joe Leffler, a postdoc in the lab, have helped with some of the recent multi-electrode array recordings, Julia Litz as well, and Lily. They have been such a pleasure to work with, and they're all gonna you're gonna see their names in future papers to come. Okay, so with that, we're talking today about both image forming and non-image forming vision. So I'll start with a very simplified schematic of the retina through a cross section. And we can see the retina is a layered structure with photoreceptors at the outermost region of the retina, closest to the outside of the eye. Light travels through the retina before it hits the photoreceptors. And then the information is photons are transformed into a synaptic signal in the outer plexiform layer, the first synaptic layer here. Then this excitatory information is transmitted through bipolar cells down into the inner plexiform layer. And then ganglion cells, which I'll focus on today, transmit this signal through the optic nerve to the brain. Now, ganglion cells come in multiple flavors and multiple subtypes, and they inherit these flavors from their dendritic lamination within the inner plexiform layer. So, for example, ganglion cells that have their dendrites in the off layer will receive inputs from bipolar cells that respond to decreases in illumination. Ganglion cells that have their dendrites in the on layer will inherit their signals from bipolar cells that respond to increases in illumination. And this information is passed to the brain to different retina recipient regions. So image forming vision is primarily driven by information to the lateral geniculate nucleus, which then passes on to the visual cortex. This underlies perception. And there's multiple types of non-image forming vision. One of these are sort of reflexive non-image forming visions. And these are mediated from ganglion cells that project to accessory optic and tactile regions, such as direction selective retinal ganglion cells. And these regions involve image stabilization and head and eye movements, amongst some others. And the other type of non-image forming vision is mediated through ipRGCs. And these are called ipRGCs because they're intrinsically photosensitive. So the IP is intrinsically photosensitive. These ipRGCs contain their own photopigment, melanopsin, and they project primarily to the suprachiasmatic nucleus underlying circadian entrainment. They also project to the olivary pretectal nucleus underlying pupillary light reflex, and they underlie many other behaviors such as feeding, body temperature, sleep, mood, and many others. So most of the image forming vision comes from the photoreceptors, which is transmitted through bipolar cells to retinal ganglion cells. But intrinsically photosensitive ganglion cells also contain melanopsin, and so they directly respond to light as well. But they're also retinal ganglion cells, so they do receive inputs from rod and cone bipolar cells. And we'll discuss some of the recent work that we've done looking at these responses in the mouse. Okay, so neuronal diversity underlies visual feature extraction. So the retina is a complex structure with many neuronal subtypes. And this diversity underlies the ability of the retina to split the visual scene into multiple parallel channels. And these parallel channels can each encode a unique aspect of vision. So fast forward to 2022, and this is the mouse retina and our current understanding of the different retinal subtypes. What you can see here is there are 3 types of photoreceptors, 1 type of horizontal cell, and then the visual information is first split up by the bipolar cells before being transmitted to the inflexiform layer, where there's huge diversity of inhibitory interneurons shapes these visual features to produce roughly 40 types of retinal ganglion cells. And each of these retinal ganglion cell subtypes encodes a unique feature of the visual scene. So ganglion cell density is concentrated in unique regions of visual space. And across the animal kingdom, different species tailor this distribution to align with their unique ecological needs and behaviors. The primate retina concentrates ganglion cells within the fovea, a small central pit in the retina that allows for acute and detailed vision. This dense aggregation of cells enables primates, including humans, to have exceptional visual acuity in the central field of view. And this facilitates tasks such as discerning fine details in our environment. Contrastingly, there's many terrestrial animals exhibit a different adaptation according to their ecological needs. Some of these include a visual streak, such as animals like the wallaby or the agouti or rabbit retina. And this arrangement optimizes the concentration of ganglion cells across the horizontal region of visual space, allowing them to scan the horizon and provides a strategic advantage for detecting predators. Now, these ganglion cells also maximize their efficiency of visual encoding by forming subtypes of mosaics, by forming mosaics and tiling across the retina. And this is an early example of retinal ganglion cell tiling with parasol retinal ganglion cells that are evenly spaced across the retina. And work from Brian Boycott and Heinz Wessler showed that these alpha retinal ganglion cells, which are the cat version of the parasol, come in different subtypes. These on-alpha ganglion cells respond to increases in illumination, and these off-alpha ganglion cells respond to decreases in illumination. And these mosaics form these territorial tilings to uniquely optimize the encoding of visual space. And these mosaics are also present in functional subtypes of ganglion cells. And so if we look at a multi-electrode array recording represented by this region here, we can see that different types of retinal ganglion cells differentially encode light to a single spot of light here. So for example, we have ganglion cells that respond to the onset of the spot, ganglion cells that are transient and sustained, and ganglion cells that respond to the offset of the light. And if their receptive fields are mapped, like they were beautifully done by De Vries and Baylor in rabbit retina, we can see that each of these subtypes of cells uniquely forms a spatial mosaic across the retina. And this is one of the first really nice examples of a functional mosaic mapping across a mammalian retina. So now we focus on the local edge detectors down here. These functional subtypes correspond to the dendritic trees where they receive all their synaptic inputs. And each of these cells uniquely encodes a subregion of visual space. And this is also present in other cell types, such as on-direction selective ganglion cells, where each of the direction selective cells responds to a different different preferred direction in visual space. For example, these two cells here that are tiling, two on-direction selective ganglion cells that respond to the same preferred direction of motion. Whereas these two cells here that have been filled with different Alexa dyes overlap visual space considerably and respond to different preferred directions. So if, for example, in these direction selective ganglion cells, we map their different preferred directions and then record the position of these cells mapped throughout visual space, we can see that cells responding to the same preferred direction form a retinal mosaic. And cells that respond to a different preferred direction form a different retinal mosaic, which overlap with each other. And this is the third direction here. So in mouse, visual channels are localized to distinct regions of visual space. Now, the mouse retina has a lot lower density than human, but their retinal ganglion cells are mostly concentrated in this dorsal region of visual space. And different retinal ganglion cell subtypes encode different features of the visual scene and are encountered at different densities across the mouse retina. So for here the on alpha or M4 retinal ganglion cell encodes information in the dorsal temporal region of visual space. Whereas the W3 retinal ganglion cell, which is thought to be involved in predator detection, has the higher density in ventral regions of visual space looking up. Intrinsically photosensitive ganglion cells, on the other hand, have their highest density in the dorsal region of retina, and this is something that we've become interested in recently in my lab. So ipRGCs are present across the entire retina, but for some reason there's an increased density of cells looking down onto the ground. And so mice have this unique distribution of photoreceptors where green photoreceptors dominate the dorsal retina or ventral vision looking down, and UV photoreceptors dominate the ventral retina looking up. And here's a really nice example from Wei Li's lab and some earlier papers describing this UV cone distribution, showing that mouse UV opsin is located in the ventral retina. Green cones, green-only cones are in the dorsal retina. Mixed green and UV cones are in the ventral retina, and then UV-only cones are also located in the ventral retina. Now, this sort of unique dorsal ventral distribution of retinal neurons is not unique to mice. It's also present in other animals. For a really good review of this, you can look at this review here. But here's an example of some somatostatin positive amacrine cells in mouse and in rabbit retina that are only located in the ventral regions. And this paper from Segar and Marshall showed that very similarly, these somatostatin retinal ganglion cells are only located in the ventral retina of humans. So back to mice, UV cones are thought to preferentially encode dark contrast, and this is important for predator detection. UV green contrast is richer in the upper visual scene or the ventral retina. And these retinal specializations match the visual scene statistics. So why is this important for ipRGCs? This is what we're really interested in understanding. We've recently found a subtype of retinal ganglion cells in the dorsal retina, and these intrinsically photosensitive retinal ganglion cells are in this region of retina that's low in UV cone opsin. And why might this be? Well, we don't really know, but this nice paper here showed that under different lighting conditions, the differences in intensity between the dorsal and ventral retina shift. So in bright daylight, the intensity differences between the dorsal and ventral retina are relatively uniform. But as we shift to lower light levels, where nocturnal animals are out foraging, the ventral visual field, or the dorsal retina, encounters a lot less luminance, and particularly at really low light levels. So these retinal specializations might underlie sort of unique adaptations that are important for encoding luminance at low light levels in nocturnal animals. Okay, so we're interested in ipRGCs. What are ipRGCs? Well, ipRGCs were first discovered by David Burson and colleagues and Sivyer Hatta, and two unique papers in Science that I'll talk about on this slide. But first, David Burson's lab injected a tracer dye into the suprachiasmatic nucleus, and then retrolabeled cells in the retina. They filled these cells and found they had the sparse dendritic trees. And then they recorded light responses to these cells. But what they did is they demonstrated that the light response persisted in the presence of cobalt and synaptic blockers to block synaptic transmission. And then they went one step further and got light responses by actually pulling the cell out of the retina and recording isolated light responses from the soma. And Sivyer Hatta and Qinghua Yao's lab made transgenic mouse lines where they inserted LAC-Z into the melanopsin locus and went the other way, labeling beautifully ipRGCs in the retina. Here we can see their axons running into the optic nerve. And then you could see their axon terminals in the suprachiasmatic nucleus and other areas such as the lateral geniculate nucleus. This is the intergeniculate leaflet and pretectal regions. And so we now know that if we ablate ipRGCs, this light-mediated entrainment, or this wheel running activity that's suppressed by light in nocturnal animals, this entrainment is abolished. And we also know through ablation of melanopsin ganglion cells that the pupillary light response is severely attenuated. And through other work, we know that if we just ablate melanopsin, circadian entrainment remains intact. And this is because the ipRGCs get rod and cone input from the retina. And so this paper looks at rod inputs to ipRGCs. So we now know that there's a huge diversity in intrinsically photosensitive retinal ganglion cells. And there's about 6 different subtypes of these cells. The M1s and M2s differ in their stratification throughout the retina and their central projections. And there's also these other morphological types and physiological types that have been shown recently. So we became interested in ipRGCs through a chance discovery by looking at a transgenic mouse line that expresses Cre under the glycine transporter 2 promoter. And this is a back transgenic. And so here's just an example of TD tomato expression in the internuclear layer showing these amacrine cells labeled. We crossed this to a rainbow line and found that there's many different subtypes of amacrine cells labeled in this line. However, when Mike was first trying to record from some of these cells, we noticed that in this seemingly inhibitory line, there were retinal ganglion cell axons traveling down to the optic nerve, which was really interesting to us. We were sort of dumbfounded by seeing ganglion cells in an inhibitory marker line. And we also noticed that these axons were only present in the dorsal region of the retina. So Mike did some really nice experiments where he injected a Cree-dependent EGFP into the eye. And we can see that really nicely labeled ganglion cells, axons going into the optic nerve, and they're only in the dorsal retina. And we also see some of these A2 amacrine cells, a glycinergic amacrine cell, labeled with this virus as well. So then Mike went and looked in central projections to see where these cells project, and saw that the axons projected to regions that are involved in circadian entrainment, such as the intergeniculate leaflet and the suprachiasmatic nucleus. And then went back to the retina and stained it with an antibody against melanopsin. And you can beautifully see here that there's retinal ganglion cells labeled with TD tomato and melanopsin antibody staining. So then Mike went and targeted these cells in the GlyT2 TD tomato line and made neurobiotin fills from them, and found that there's two principal subtypes, which are the M1 and M2 cells, which differ based on the stratification in the retina. And here's another nice example of the different dendritic morphology. He did some analysis of shoal analysis of the dendritic crossings and showed the two main subtypes differed significantly in their dendritic morphology. And he also looked at the dendrites of M1 ipRGCs in a different mouse line that ablates most ipRGCs except the M1 subtype. And so these ipRGCs were indistinguishable from the M1 subtypes that were found in the GlyT2 Cree line. So then we went and after neurobiotin recordings, targeted these cells, neurobiotin fills, targeted these cells for light evoked recordings. And we can see here some light responses in current client recordings in control. And then after adding synaptic blockers, excitatory blockers, we see these really nice intrinsic photo responses. And so what we found really interesting was we can see that there's many of these ipRGCs that are also labeled with TD tomato in this line, but there's also a bunch of ipRGCs that are unlabeled and overlapping. So we thought, well, maybe this represents a unique subtype of ipRGCs, and maybe they form their own mosaic. And this was somewhat inspired by earlier work from David Burson's lab, showing that ipRGCs, if they trace their dendrites, overlap considerably. And so the coverage factor, the coverage factor indicates the number of ipRGC subtypes or number of ipRGCs that cover a unique region of visual space is about 3.8 for M1 ipRGCs and 4.6 for M2 ipRGCs. And because we know that the average coverage factor for a retinal ganglion cell subtype is around 2, This made us think that perhaps there are unique subtypes of ipRGCs forming their own retinal mosaics. So Mike went and painstakingly mapped these ipRGCs across the entire retina in multiple animals, and showed that the GlyT2 ipRGCs are only present in the dorsal retina, and this overlaps with a region of visual space that's low in cone density. He then went and looked at the melanopsin immunostaining in each of these cells and followed the dendrites down into the off layer, or in other ipRGCs, followed their dendrites into the on layer. And so here he could uniquely identify which of these ipRGCs were M1s or M2s. And so what's really interesting here is that if we look at the distribution of these across multiple animals in the dorsal and ventral retina, if we subtract the ipRGCs that are in the dorsal retina, then the number of ipRGCs, or their coverage, is even. So this is further evidence from looking at their mosaics that ipRGCs in the dorsal retina, labeled in this line, form a unique subtype that's restricted to this area. Okay, so another way to look at this is, now that we know there are certain M1 ipRGCs and they project to unique regions of the hypothalamus, we thought that if we go the other way and look at the central projections of these cells, we might be able to retro-label them and differentially label M1s and M2 subtypes. So the central projections of these cells are really interesting. They project to the suprachiasmatic nucleus, but a unique sort of shell region of the suprachiasmatic nucleus. But we also notice that the projections of these ipRGCs form a really dense input to a region called the supraoptic nucleus. And the supraoptic nucleus is a region, in addition to the paraventricular nucleus, that sends projections to the pituitary, where they release oxytocin and vasopressin. And if we label the supraoptic nucleus with an antibody against vasopressin here, you can see this is the supraoptic nucleus here and the paraventricular nucleus here. So we target our injections to this region of the supraoptic nucleus to see if we could retrolabel these cells. And so here's our injections. We injected in the supraoptic nucleus and then looked back in the retina. And what we saw was striking. Now we can label only the M1 subtypes of ipRGCs. And these ipRGCs form a mosaic, a regular tiling mosaic across the retina. And they're only restricted to the dorsal retina. And this is in the GlyT2 line. So next, we injected into the supraoptic nucleus in a different transgenic line. So we reasoned, is there something about this GlyT2 line that's restricting these retinal ganglion cells to the dorsal retina? So now we use OPN4 to restrict the expression of cells in the retina. We reason that if we see the same population of cells, then it's likely that this supraoptic nucleus population is a unique population of cells restricted to the dorsal retina. And so Mike did this beautiful work here, where even though the injection's in a completely different Cree line, most of the cells are restricted to the dorsal retina. We do see some sparse labeling in the ventral retina in this line. But in the OPN4 Cree, we see a unique mosaic of cells. Their somas are spaced regularly, and they tile the retina. And this is some quantification. Okay, so I'm going to skip the summary. So do these ipRGCs release GABA or glycine? So we know from recent work from Tiffany Schmidt's lab that there's a GABAergic population of ipRGCs. And because we found these cells in a glycinergic line, we wanted to know whether they're releasing GABA in the SCN or the IGL. So Misha Moldovan from Chuck Allen's lab recorded from the suprachiasmatic nucleus in a GlyT2-Cre AO32 mouse where channel rhodopsin is expressed with the GlyT2-Cre mouse line, and found that after adding either strychnine or gabazine, there was hardly any change in the amplitude of the light evoked response. But adding CNQX and AP5 to block excitatory transmission, it completely abolished the response. And so we also took another approach where we injected an AAV flexed channel rhodopsin into the eye to try and restrict the channel rhodopsin specifically to the retina and their retinal axons, and then went and recorded from the intergeniculate leaflet. And Tavita Garrett did these recordings, and in collaboration with Mark Meadows and Ricky von Gersthoff, and found the exact same response, exact same result, where light evoked or channel rhodopsin evoked synaptic currents were completely abolished by excitatory blockers. Okay, so it seems so far that these GlyT2-Cre ipRGCs are glutamatergic, at least from our recordings in the SCN and the IGL. So next, we're interested in whether these cells express the GlyT2 or SLC6A5 gene. So one of the ways we're going to address this is using intersectional genetic lines where we cross the GlyT2-Cre with a VGlut2 FLIPO and other intersectional inhibitory lines. And Tavita is also doing this really nice work with help from Catherine Morgans to perform RNA scope in whole mount regions of retina. And this is the expression of OPN4 mRNA throughout the entire retina. And so far, Tavita has been doing OPN4 and GlyT2, and really has found no evidence that these ipRGCs express any levels of GlyT2 mRNA. So it seems like they're glutamatergic. Okay, so the next part of my talk is going to talk about recent work in the lab to look at the functional differences in these cells using MEA recordings. Mike Berry's using a 256 channel multi-electrode array here and putting the retinal ganglion cell layer down onto this array. And the reason we did this is it allows us to record light responses from the cells prior to stimulating the retina with any fluorescent tags, which might adapt the retina or adapt the melanopsin. And so we also wanted to exploit the difference in melanopsin expression between different subtypes of ipRGCs. M1 ipRGCs express a lot of melanopsin and have large intrinsic photoresponses, whereas other ipRGCs have lower levels of melanopsin and have smaller intrinsic photoresponses. And so in the MEA recordings, we reason that we can try and isolate these different responses and tease out this diversity in ipRGCs. And even within M1 ipRGC subtypes, there's diversity. Different M1 subtypes project to different central regions of the brain, underlying differential responses. Some really nice work from Michael Doe's lab has shown that M1 ipRGCs differ in their intrinsic responses and encode different levels of light based on their spiking outputs. And work from Tiffany Schmidt's lab has shown that the M1 ipRGCs differ in their photoreceptor inputs. So in our experimental paradigm, what we do is at first we record light responses from all retinal ganglion cells on the multi-electrode array and present a diverse set of visual stimuli that we can record and look at later. Next, we block synaptic transmission and pulse blue light at different light intensities. And this allows us to uniquely tag ipRGCs. We get really nice light responses from ipRGCs on the MEA. And a really nice signal to noise where we can isolate each of these cells when we're doing clustering. And then Mike used this really cool new technique where he adds an opsinamide and then uses a really bright 18 hertz flicker frequency. And this is used in mice where GlyT2 Cre drives the expression of channel rhodopsin. And so now we're assuming that ipRGCs that we've tagged through their intrinsic photoresponses and respond to this channel rhodopsin response optotag cells. And here are some of the optotag responses in spike rasters. Here are the optotag cells here and other ipRGCs here. And so then we can take these recordings, find optotag cells, go back to these groups, and then look individually at their light responses to either photoreceptor drive or intrinsic photoresponses. Okay. We also did unbiased cluster analysis of the light responses, similar to Tom Barden's lab. Tom Barden's work in Thomas Euler's lab in 2016. And here we did cluster analysis where we looked at the principal components that explain greater than 5% of the variance in their responses. And then this minimal Bayesian interference criterion gave us 8 clusters of ipRGCs. And here they're organized according to their intrinsic photosensitivity. So now we're assuming that these most sensitive clusters are the M1 ipRGCs, and then these less sensitive clusters are different subtypes. And we broke these clusters up into 3 groups, differing based on their sensitivity to light. There's the high sensitivity groups, which contains a sustained and a transient response, the medium and the low sensitivity groups. So these have really different responses to low light levels. We can see the high sensitivity groups are here, the medium and the low. And this is showing that the average firing rates sort of split between the high sensitivity groups. And when we look at their time to peak, the highest sensitivity groups are the fastest to respond, and that this changes over different groups depending on their sensitivity to intrinsic responses. But these first 2 clusters that are the highest sensitivity differed really significantly in their response duration, with the highest sensitivity group, which contain the most optotag cells, being the most sustained of the ipRGCs. Whereas the most transient of the ipRGCs was also one of the high sensitivity groups. And this is just some grouping data showing that most of the optotag clusters, ipRGCs fell into this first cluster. Okay, so we think that these ipRGCs are M1 ipRGCs. And these 2 subtypes, one of these, which we think is the superoptic nucleus projecting cell, has very sustained responses and will respond to illuminance for a long duration, up to a minute. Whereas the other ipRGCs, which are not present in our optotag group, are very transient. And we think these are likely the monitotal and unimodal ipRGCs that were described by Michael Doe's group, where the unimodal cells go into depolarization block at higher light levels. So Mike then went back in time and looked at the light responses of these ipRGCs before blocking synaptic transmission. And then here we're just looking over the 4 sort of high and medium sensitive clusters. And what's really interesting here is that their light responses to low light levels, at least to these sort of green sustained visual stimuli, don't really differ that much. Most of the diversity in their response occurs at higher light levels. And Mike also went back and performed the same unbiased cluster analysis, but now including both the intrinsic photoreceptor responses and photoreceptor-derived responses. And the really interesting thing here is that now the number of clusters, which we're calling groups, decreased to 6. And so Mike went and organized these cells according to their groups from highest to lowest sensitivity, and then which of these groups contain the intrinsic photosensitive clusters. And the next thing we want to do is see if we can map these clusters to the known molecular ipRGCs that have been described by the Sains lab. And so we think these group A and B are likely the M1 ipRGCs that are most sensitive. We think this group C is probably an M2. However, group D and some of these other groups are a little less certain. And so we're now doing some single cell recordings to sort of shore up this data. And what was really interesting is that when Mike plotted the photoreceptor responses at different light levels, he found that most of these ipRGCs respond really well to photoreceptor-derived input in low light. However, when these stimuli are present at photopic light intensities, their contrast responses become severely attenuated. And so here's the ipRGC groups from A to F. And this is compared to an on-transient retinal ganglion cells that's a non-ipRGC. And we can see they have relatively nice contrast responses from low to high in scotopic and mesopic. However, the contrast spike frequency modulation is severely attenuated at photopic light intensities. And we think this is because melanopsin's depolarizing the cells and sort of washing out the photoreceptor-derived spike input. And this was also present in their frequency responses. We can see that the different subtypes have differential frequency encoding, with some of these lower sensitivity groups having really good photoreceptor-derived frequency encoding. And so we're interested to follow up on this, given this really nice previous work from Tiffany Schmidt and Sama Hatter's group showing that melanopsin enhances contrast responses. So now, for the future directions of the lab, we're really interested in what is the whole meaning of having these unique ipRGCs in the dorsal retina, looking at ground luminance. And there's a really cool study by Wolf and Summerlin that looked at the activity of mice in an open arena in their natural environment. They just built a cage around sort of their feeding habitats. And then they track their activity by marking sort of marking grids in the sand and measuring crossings in the grid based on where the mice were. And they looked at whether the mice were out feeding from food that was provided in the open, or feeding under cover, and basically showed that mice have quite significant activity at night, uncovered, but as soon as there's any proportion of moonlight out, which is represented by these different areas here, their activity is suppressed. And so perhaps light that's reflected off the ground in nocturnal animals is important for behaviors that drive the avoidance of predation, so that they can go out and forage when the light is low enough to where they'll be sort of covered by lack of light. And so we're now designing these cages that's spearheaded by a student lab, RISPA, where we can control the light presented to the animals in the ventral and dorsal regions of the visual field, while we track using video monitoring and sensors, where we can sort of track their running activity and feeding activity, drinking, and sort of home cage activity. So that's the future directions. Thanks for listening. I just want to acknowledge all the members of the lab who've worked on this, particularly Mike Berry, Tavita Garrett, Mark Meadows and Enrique von Gerstol's lab, and Misha Moldovan in Lane Brown's lab. I also want to acknowledge Lane Brown for helping us out with delivering transgenic mice to us. And I'll stop there.
Thank you very much, Ben, for this very interesting and thought-provoking talk. So much information, and I have so many questions of mine. There are a lot of people following live, and given that there currently are no questions, I'm sure that some of them want to ask offline. So I already posted the Zoom room link there. We are waiting for some questions to appear, but in the meantime, I have a lot of questions of mine. So I will start from the future directions and maybe take you back at the beginning of your presentation as well. So I take it that you believe that melanopsin expressing ganglion cells play a bigger role in nocturnal animals versus diurnal. And I was wondering whether you have any ideas or assumption or speculation about different subtypes, let's say in squirrels that are diurnal and have like cone-dominated retinas and not so much rod-dominated ones.
Yeah, that's a really good question. I think my assumption would be that nocturnal animals have increased melanopsin sensitivity and possibly more melanopsin subtypes and increased processing power because there's less light. And when they're in their burrows at night and when they're in their burrows in the day and they're sleeping, they probably don't need to use these circuits. So that's sort of the working hypothesis for us at the moment, is that melanopsin is a little more sensitive in nocturnal animals.
Right, and I take it that it's also like more comparable in kinetics with the rod responses than with the cones. So probably you could use it together, like the signal that you get from the typical photoreceptors and their own intrinsic signal in terms of latency and
it's really interesting what the differences in rod and cone-dominated retinas would be to ipRGCs. I think mice have pretty strong rod inputs, as they do to all of their ganglion cells. But yeah, it'd be really cool to look at diurnal animals and see what their sensitivities are. I bet they're just less sensitive because the cones have a higher threshold. So, yeah.
There are already congratulatory messages appearing in the chat. I'm conveying them as well because you cannot see the chat, but they will appear there later on as well. And another question, I think like also general one in that case, would be, so like there was a recent Christian Puller preprint about displaced ganglion cells. And if I'm not mistaken, like I think it's in bioRxiv currently, so they show that there are also displaced M1 ganglion cells that have like this dorsal density. I was wondering in your glycinergic line, whether you observed any displaced cells maybe in the inner nuclear layer?
No, actually, we don't. We see them overlapping with displaced cells, but we don't see any of our glycinergic cells, TD tomato or GFP expressing cells that are displaced.
Right.
Yeah.
Okay.
So we think they're definitely separate subtype to those.
Thank you very much for addressing this as well. One question that appeared now in the chat is from Sivyer Schroeder. Maybe naive question. My impression was that ipRGCs provide information about luminance levels, so I was surprised to see so much, and in parentheses, and fast adaptation. What are your thoughts?
So much, fast adaptation?
Yeah, so much adaptation and so fast adaptation as well.
So fast adaptation, yeah. Yeah, it's sort of interesting. I think a lot of this adaptation and fast adaptation in some of the subtypes is really spike frequency dependent adaptation. That's not necessarily melanopsin adapting. It's the spiking response of the cell due to depolarization block, and this was shown by Michael Doe's group in some of the traces I was showing from Milner and Doe. It's unclear if this depolarization block or spike frequency dependent adaptation changes across different light levels, but we do see some really sustained cells that can perform sustained firing over long time frames, and there's this unique difference between the sustained and transient cells that we're seeing in the most sensitive groups. And we think that most of this adaptation is spike frequency dependent.
Along these lines, I was wondering, so like at the chirp part where you do contrast modulations, what was the temporal frequency you chose for the contrast modulation?
1 Hertz.
1 Hertz. And they could easily encode 1 Hertz at the temporal chirp as well, right? So your temporal chirp starts from 1 and goes up to 8 or 10?
The temporal chirp starts from 0.5 and goes to 16.
Right, okay.
Yep.
And all clusters gave up at a similar point, or you had some clusters that could follow up to 10 Hertz or 16 even?
The lower sensitivity clusters follow higher frequencies, and that's sort of what we'd expect. The lower sensitivity cells we think are maybe the M4s, the M6s, although we're not really sure exactly which ones. But we were super surprised that the most sensitive group that we think are the optotag cells in the dorsal retina also had really nice photoreceptor responses and followed the sort of contrast modulation similar to some of these low sensitivity groups. So that was surprising. Also surprising that the temporal frequency and contrast modulation was inhibited by melanopsin activation, which was sort of unexpected. We have maybe some thoughts about what that is. We can discuss that in the next session.
Right. I mean, given that you say that, and among the other congratulatory messages, I will go ahead and post the Zoom room link so we can also continue offline with some questions in case there is interest from the people sitting in the audience. And like one last question I had is like, because you said that you hold this assumption that these groups correspond to M1 because of the action potential and so on. Is this a safe assumption? Like, it's a very naive question from my end.
Yeah, it's a good question. No, it's not a safe assumption. I don't think we can make assumptions, and we really need to go and make single cell recordings to the same light stimuli and then put them in our clusters. Yeah.
Okay. Given that people are already joining us here in Zoom, I would like to thank you once again, Ben, for this fantastic talk, and thank the audience as well for attending yet another Sussex Vision seminar. Thank you.
Thanks so much, George.